Hey everybody, this is our spectrophotometer lab, and in this lab we will be demonstrating the Beer-Lambert law, which is essentially the law that describes the linear relationship between the concentration and the absorbance of a solution. So by now the dilutions have been made, and next you go to the computer and hit 0 so that you can load your blank solution. So right here, we are just loading the blank, and we start off by cleaning both sides of the cuvette with a chem wipe and closing the door and loading it. From there, we load our stock solution, and of course, after wiping with a chem wipe and closing the door and getting a read on that as well. As you can see, we're just continuing that same process of wiping the cuvette and loading them to get the reads. And we basically just do each dilution from there until all the dilutions are complete. Remember, it is important to zero the spectrophotometers for readability and accuracy, and we also made sure to scan across multiple wavelengths so that we can determine the concentration of the absorbing species and also identify unknown substances. We also scan across multiple wavelengths because of the lambda max, and the lambda max is the wavelength at which a substance has its strongest photon absorption, which is why we scan at these multiple wavelengths. In order to find the lambda max, we have to scan at these multiple wavelengths in order to compare the different wavelengths and determine which one is the strongest or has the strongest absorption. Make sure to record all of your data samples in your lab notebook so that you can later determine the relationship between the concentration and the absorbance of your dilutions. After doing all of your scanning and retrieving your data samples, you should find that the relationship between the concentration of the solution and the absorbance of the solution is directly proportional.